And as a neurogeneticist and as a physician scientist, my overarching goal is to improve both the diagnostic part and then move us towards the treatment part for neuropsychiatric disorders. And especially in these rare disorders that are monogenic, we have a lot of gaps in what we know. So that starts with understanding what are the symptoms that each individual has. But we can do this through a lot of different natural history study approaches. We have to collect a lot of data, genetic, about how people are feeling their symptoms. And then we move this into a variety of different types of model organism systems. And I'm going to be talking about one type of model system, which is doing iPS or stem cell-based neural organoids. Then we have to understand why these disorders happen, with the long-term goal of being able to come back to a treatment. Years ago, Dr. Yamanaka let's see if that works, there we go, first pioneered being able to take skin cells, so I can do a skin cell biopsy. Those can get reprogrammed into stem cells. And then Dr. Pashka, who I work with, and others started to understand how can we try to help these stem cells differentiate into different parts of the body, such as, for example, how do we make them become more like neurons or the cells in our brain? And initially, these were grown in a dish on a 2D flat surface, which is really helpful if you want to study a particular cell type that you can make, or if you want to be able to study drugs that you want to apply just to these cells that are flat. But our brains are not flat. Our brains are three-dimensional with a lot of complexity. And so it turns out that if you take these stem cells and you encourage them to group up together, they will, on their own, start to form, with a little bit of help, they'll start to form organoids. In other words, an organ-like spheroid. And if we give them certain chemical mixes, we can encourage them to become like different parts of the body. We can encourage them to become like the brain and like different subregions of the brain. And then we're able to grow them in dishes like this, where they're free-floating. And while nothing will mimic the human brain exactly, this at least allows us to have a little bit of that 3D architecture, which means that we're able to have different cell types, mimic different regions of the brain, and then mimic some of the circuits that will be there. But the human brain is even more complex than that. And so we can take different organoids and we can give them different cellular signals, different chemical signals, to make them like different parts of the brain. And then we can encourage them to fuse together or assemble together into what we call assembloids. And then we can watch here if one on the left is the surface of the brain and the one on the right is the deep parts of the brain. We can watch our interneurons move from one into the other. And this means that we can study some of the more complex interactions between different parts of the brain, how these areas wire and fire together. And these are some of the types of techniques that I'll talk about in a moment. We're able to use these types of model systems in order to look at electrophysiology, the electrical excitement that we have in our brain cells. We can look at them under live imaging. We can watch the cells in real time as they move around or connect. We can look at their anatomy. How do they connect together? Where do those cells go? And we can also do all of the different types of omics, all of the complex single cell or multi-organism level studies of how our genetic information and the proteins and parts of the cell are modified in disorders. And then I've just described to you most of the left part of this image. And all the way over at the right is one of the new things that we've been studying in the Pashka lab, which is we know that looking at cells and organoids in a dish is helpful, but what happens as we get closer and closer to what happens inside of our bodies? And so if we make these neural organoids, we can actually transplant them into the brain of young rodents. We can let them integrate there so that they're getting more of the signals that happen in a living body. And that means that then we're able to understand how these can be involved in circuits in which we're getting information from the environment, and we're able to see behavioral outputs, which gets us a little bit closer to understanding what might happen if we wanted to understand human cells from a human disease in a human tissue to try to make a treatment that will be best suited for humans. And so next I'm going to describe a little bit about how we use these different models in order to study one particular disorder that's involved in autism. And we're going to talk a little bit about calcium. The gene that we're going to talk about today is called CACNA-A1C. And it encodes one of the calcium channels that has the delightfully complex name of CAV1.2. And what happens is that this is like a little pore, like a revolving door in our cells. And what it does is it lets calcium come in and out, or mostly in, actually one way. And calcium is really complicated, and this image is complicated on purpose, and it's just a fraction of what calcium does inside of the cells of our body and in the cells of our brain. And calcium is kind of like Goldilocks. It wants to be just right. Too much of it, too little of it, and we're going to have problems. And inside of the brain, calcium is heavily involved in how cells move around and how they respond to action potentials, which are the exciting electrical signals and impulses that we have. And calcium is also very important in the heart. And that gene I said, CACNA-A1C, it turns out that when you have different changes in it, you have what's actually a spectrum of disorders that we call CACNA-A1C-related disorders that mostly involve the heart, the brain, sometimes both, and sometimes other parts of the body. And in the heart, if it's expressed just in the heart, we have arrhythmias like long QT syndrome. We have short QT syndrome. Sometimes there's changes in the structure of the heart. But in Timothy syndrome, which is what I'll be talking about for the rest of this, which is a subtype of CACNA-A1C, not only do we have the heart rhythm problems, there's differences in facial features, there's syndactyly, which is webbing of the fingers, and then there's incredibly high rates of autism, epilepsy, and intellectual disability. And Timothy syndrome type 1 and type 2, they describe two different genetic changes in the same area of the gene, both of which are a gain of function, too much calcium getting in. But then we started learning, as more and more people were having genetic testing, that there can also be a neurologic-only phenotype, only the brain and mind. And in this case, autism, epilepsy, intellectual delay, and disability are incredibly high in their rates, but the heart seems to be healthy. And then what I just described to you are what we call pathogenic variants, something that directly impacts your health. But it turns out that common variants in this gene are also some of the highest hits when we look at population-level studies of autism, bipolar, depression, schizophrenia. So clearly this gene is involved in our health, whether or not we have a common or rare pathogenic variants. And with the help of Catherine Timothy, the Timothy Syndrome Foundation, the Timothy Syndrome Alliance, I was able to put together a cohort of individuals who have CRD. And first we wanted to understand how common are these neuropsychiatric phenotypes. And it turns out that across the board, no matter what someone's genetic change is, that these were very common. Autism spectrum disorder formally diagnosed in half of them, autistic symptoms in 90%. And then we said, is this because of the heart issues? Is it that their heart has an issue, it injures the brain, and that's what happens? Well, we saw no statistical significant difference between those with and without heart problems. So we know that this is really impacting the brain directly as well. That didn't work. So CRD is therefore a really interesting genetic model that we can use to try to understand what is the role of calcium? What is it doing in brain development? How does it impact our developing brain over time? And then we can take that one step further. If we think about something like Timothy syndrome, which is this gain of the channel, can we try to think about treating these neuropsychiatric symptoms? And if so, what is this therapeutic window for treatment? Could we be too late? Could it be just at the right point of time in order to make a difference? Oh, that's going backwards, all right. So as I mentioned, this TS1 variant, it's this increase in calcium. And so what happens is, and this is work that Dr. Pashka has been working on for over a decade, if we take skin cells from people with and without Timothy syndrome, we turn them into these stem cells, and then we grow them into these organoids that mimic the surface of the brain. This is a graph of what happens to the calcium levels. And what you can see is that in red, those who come from individuals with Timothy syndrome, they have more calcium. And we can actually block that if we use some of the types of medicines that block calcium channels in the heart. But the problem is that these don't enter into the brain. They can't pass the blood-brain barrier, and certainly not at levels at which it would be safe for them to be used for the heart. So we need other approaches. And in this case, what we've done is we've taken those stem cells, we've turned them into organoids, and we've turned them into organoids, some that are on the surface of the brain and some that are deep in the brain. And when you do that, you can actually watch those interneurons migrate from the organoid at the bottom, and they're moving their way up to the top. You can see them in live time. You can follow those arrows. And when we compare those neurons in people with and without Timothy syndrome's on the bottom here, you can see that the Timothy syndrome interneurons, while they're jumping quickly, they're not moving very far. And that's shown here in blue, that overall they don't seem to migrate as far over time. And so now we have an idea of some of the phenotypes or differences that we can see in these organoids in a dish, and we wanted to know, what could we do in order to change this? What are some of the approaches? And TS1, not only is it a gain of that channel function, more of that calcium coming in, but it turns out that it also affects splicing. And so splicing is what happens. Our DNA is this long string. Here we have exons that are shown as the rectangles, and the spaces in between are known as introns. That's the line in between. And what happens is that those have to get cut and pasted together. And with Timothy syndrome, what happens is not only does it cause an increase in function, but it also increases its own splicing, so it amplifies itself. It's like a good self-promoter. And that happens in which we have more of this exon 8A and then less of this exon 8 that you're seeing in the blue in the graph. And I'm very thankful that Dr. Brent already told us a little bit about antisense oligonucleotides. What these do is that they modulate RNA. They do it in three main ways. One is RNA degradation. If you make an antisense against RNA, it can lead it to get broken down over time. They can also block translation. So in other words, the RNA can't be made into a protein. And finally, they can change splicing. And this is what we wanted to use it for. So you can either cause it to put something in to keep it, or you can cause it to take it out. And Xiaoyu Chen, who's a fantastic postdoc now, research scientist in the Pasca lab, she created a variety of antisense oligonucleotides all across the area and then kind of competed them against each other to see which ones would have an impact. And she was able to find several that led to less of that 8A being included and more of the 8. So you can see less of the green on the top, but still having plenty of the exon 8 in the blue on the bottom. And when we started to give that antisense oligonucleotide to organoids and assembloids in the dish, that's shown here in purple compared to the control in tan and the TS ones that are in teal here, we're able to see an improvement or rescue in that calcium level. So the calcium level is going back to normal. And then when you do that with electrophysiology, when you listen directly to the electrical activity of the cells, which here you can see the teal is too low, and instead we're able to rescue that back up. So it's changing what happens with the calcium level, and it's changing what happens with the electricity. What else does it do? And if we go back to the idea of having the assembloid of the surface and the deeper parts of the brain regions put together and looking at those interneurons, we see that the way that they migrate also gets rescued. So they jump less frequently, they get to go a little bit further. So we're starting to see a difference in the way the cells actually are working. And then if we take these organoids made from the stem cells of people with Timothy syndrome, if we transplant them into the brain of early rodents, let them grow up, and then if we give the antisense into the brain of those rodents, and then if we look at that calcium activity in the live slice, you can see after you depolarize, the calcium activity goes up. In the Timothy syndrome, in the teal, it's very high, it stays high like we were seeing before. But when we give the antisense, we see an improvement in that calcium activity. And when we look at the actual cells that are inside of those organoids, normally the ones that are untreated with Timothy syndrome, you can see how shrunken and shriveled they are compared to the wild type that are on the far left. But when we give the antisense oligonucleotide, that arbor, those networks, the branching of the brain cells inside of the organoid are improved. And you can see that on this graph where when we look at the complexity, that purple line is going back up towards the tan. And so what we see here is that we are able to change what happens at what we call the molecular level. So we're changing what's happening with the DNA expression. We're changing what happens at the electrical level. And we're seeing a bit of a change in what is happening at the cellular level. Not only that, we're seeing something that's changing in real time as you dose the antisense to the animal with the transplanted organoid. And so we're hoping that, as Dr. Berman said, it's really important that we can move this from the stage of the dish or of the animal into human beings. And that's some of the work that we hope to have ongoing now. And so I'm just here to say that our hope is that neural organoids are another tool in the toolbox of what we can use to understand autism and other neurodevelopmental disorders. That the organoids themselves, a single organoid, could help to model more human-specific biology, look and see what's happening in human-specific diseases. When we have these assembloids put together, then we have a chance to be able to look at migration, at circuitry in a little bit more complex way. And these transplanted organoids help us to model circuit input, behavioral output, and continue to get closer and closer to something that is more like what is happening in individuals with these disorders. So with that, I want to thank, first of all, the Timothy Syndrome Foundation and Catherine Timothy, the Timothy Syndrome Alliance, and Sophie Muir, all the participating families in this work. I want to thank all of you for coming and learning about this. I want to thank the Autism Science Foundation and everyone who makes this science possible. I want to thank Xiaoyu Chen and Fikri Bure, who are leading this work that I got to be a part of, Sergio Pasca, who's been a wonderful mentor in the lab. And mentorship is so important, and we always get a chance to thank the people who we're working with right now. We don't always get a chance to thank the people who we've worked with before, and so I just want to thank Dr. State, who was actually my mentor when I was in college, and who inspired me to go into this field. And I just want to say that now is a really important time for mentorship. It's very hard to think about going into science in this day and age, and the more fantastic mentors we have out there, the more we're going to be able to continue this work. Thank you. Any questions?
Yeah, I'm curious. Sure. So my curious question is, given the presence of other regulators of intracellular calcium, what is unique about the function of the CAV 1.2 channel that allows it to have the phenotype? Is it cell-specific expression? Have you narrowed that down?
Fantastic question. Do I need to repeat, or was everyone able to hear? Sounds like everyone heard. So you're right, there is a lot of overlap. There are several different types, as you can guess by the fact that there were so many numbers after it said the calcium channel, that there are many different subtypes of calcium channels, ways in which they're kind of almost like a Lego set, kind of pieced together. And we're still trying to understand what is it about this one in particular. We know that there are other phenotypes that happen with other calcium channels, but this one seems to be particularly strong and unique phenotype. And as you mentioned, we think it has to do with when and where this calcium channel is expressed. It is very predominant in the brain, also in the heart. But we're also learning from some fantastic work out of a group in the UK that even this, I talked about that splicing of the Timothy syndrome area versus not, but this gene actually seems to have about 240 spliceoforms. So all over our body, it's being modulated in different ways. And we think that that might be part of why it has so many impacts, not only all over the body, but why the impacts are so strong in the brain in particular, is because probably it's the regulation of it is quite complex, and therefore it's having even more of an impact on what's happening. Because these calcium channels have a lot of similarities, it means that we actually don't have any drugs right now that affect just one of them at a time. So they tend to affect a whole bunch of them in one go. And that means it's sometimes very hard for us to isolate how to help just one of those channels. And again, that Goldilocks idea to get it just right is very challenging. Yes, there's a lot of unanswered questions in that area.
Hi, my name is Eric Lontok from the Coalition for Aligning Science. Thank you very much for your talk. I have two related questions. Is the 8 and 8A splicing balance that's occurring in the brain, is that similar to what's occurring in the heart? Is there also a transition from the 8 to the 8A isoform over time?
Yeah. So in general, the 8A seems to be more expressed early, and then it tends to go down. But part of the issue here in Timothy syndrome is we get this persistent 8A. We are hoping that paper's supposed to come out any day now. I was just speaking with this team in the UK, but they've been trying to study this over time and in all different tissues of the body. So hopefully any moment now we will have some better ideas of what that regulation looks like, not only in the brain and in the heart, but also across mostly, I think, postnatal development in humans.
And so I asked that because you described both the cardiac phenotype and neurodevelopmental phenotype. So you showed beautiful data of how you're impacting, you're modifying the neurodevelopmental phenotype. And I wonder, what is the impact of that ASO in cardiac cells?
Yeah.
And whether or not, because if you're diagnosing these individuals earlier, and long QT is a condition that really affects young children, right? That's when they're most precarious, most susceptible to it. So it just, much like you could hit I guess it's the thing about syndrome, right? You want to hit multiple organs.
Yeah.
So I guess my overall question is, how is your ASO being delivered such that you might potentially hit more than just the brain?
Yeah. Yes, ideally, you'd be able to take one treatment and treat the whole body all at once. In this disorder, because of the arrhythmia, all of the children have implanted defibrillators. So we know that their heart is kind of safe, as safe as it can be, no matter what. But that doesn't help the brain at all. And they have plenty of, again, these cardiac medications can be very helpful for the heart, but they're not really passing into the brain. The way things stand with how different drugs are regulated is that you can think about trying to deliver to two spaces at once. So in order to get an antisense into the brain, you have to deliver it past the blood-brain barrier. And there is some leaking that happens back into the rest of the body, but it's something that we want to look at and try to understand better. And we have not had the chance yet to think about what would it be like to try to give both in the body and into the brain space at the same point in time, but that is something that we're very curious about. Wonderful. Thank you so much.